Hi everyone, welcome to your first super awesome science lesson. It's on the scientific method and experimental design. Starting with the scientific method. Scientific method's a way for scientists to organize their thinking. It's basically brainstorming for scientists. If scientists didn't have this way to organize their thoughts and to design their experiments, then other scientists might not be able to repeat their experiments or repeat each other's experiments, and they might not be able to come up with the same results and might not be taken seriously. And it's really important that they are taken seriously by the public because they do a lot of really important work and contribute greatly to society. The main parts of the scientific method are as follows. First of all, scientists have to make an observation. The observation's made using qualitative or quantitative data. Qualitative data refers to anything having to do with your senses, and quantitative data is anything to do with numbers. So a good example, an easy example to understand would be, it feels hot in this room. Feels hot in this room would refer to qualitative. That's a qualitative statement because you're using your senses. You're noticing that it feels hot. Whereas if I said it is 85 degrees in this room, that would be a quantitative statement because I'm using an actual number. The next thing is a hypothesis is made. A hypothesis is simply an educated guess. So why do you think it feels hot in this room? And it's usually written as an if-then statement. So we might say, if the blinds are open, then it will feel hotter in the room. Or if we just came in from HAC, then we might be hotter and the room might feel hotter. So we might do some things to design and perform an experiment. Maybe we try opening the blinds or closing the blinds or walking or cooling off before we come in from HAC. There's a number of things you can do to design and perform your experiment, and there is a specific way to design your experiment and specific variables and things that you have to include, and we'll get to that in just a minute. After you design and perform your experiment, you need to analyze and graph your data, and this is the part that tells the story from your experiment. After you have told your story through analyzing and graphing your data, you either accept or reject your original hypothesis based on whatever your results are, and then you can make your conclusion. So, accepting or rejecting doesn't mean right or wrong, and I'd like to really stress the importance of that because students often feel that if they have a rejected hypothesis that they're wrong or they failed. And that's not the case. It just means maybe you need to ask more questions, or maybe somebody else is going to come along and ask a question that you didn't think of, and you design another experiment and the process keeps going. So back to qualitative and quantitative data. Some examples, again, are smelling, touching, seeing of qualitative, anything dealing with your senses, and quantitative, anything dealing with measurements. And since we make lots of observations in science, your observations are sometimes going to be qualitative and sometimes they're going to be quantitative. We're going to be making a lot of measurements. We're going to be talking about how hot or cold something is, and we're going to be using actual numbers as well. So it's really important to understand the difference between the two. Some examples here, the book is heavy. That's a great example of a qualitative statement. It doesn't tell you how heavy the book is, you just know that the book is heavy. The formula for density is mass divided by volume. This one's tricky, and it could be argued to go both ways because the formula for density is mass divided by volume doesn't tell you any numbers. However, density is a number, mass is a number, and volume is a number. So if we did plug numbers in, then it would be quantitative. The temperature outside is 20 degrees Celsius. That would definitely be quantitative because you're using a number. And then it is warm outside is qualitative. We don't know how warm. We just know that it's warm. Okay, so when we're designing and performing our experiment, we have to keep several things in mind, and we have to include several things. First of all, we need to have an independent variable. So what is the investigator changing during the experiment? That's up to you to decide. That's the exciting part, is you get to pick it, depending on what your experiment is trying to prove. the independent variable is always measured and graphed on the x-axis. So when you graph and analyze your data, you always find this on the x-axis. The dependent variable is what is measured. So what are you measuring or what are you observing? So that's your qualitative and your quantitative right there. And that's always going to be on your y-axis. The experimental group is the group that is exposed to the independent variable. And I'll explain a little bit more about the experimental group in just a minute. The dependent and the independent variable, when they are graphed, again, the independent is on the x-axis and the dependent is on the y-axis. A great way to remember the difference between the two of these is you can say blank depends on blank. So in this case, we know that height is the dependent variable and time is independent. So you would say height depends on time. And that makes sense. That's true. Height does depend on time. As time goes on, height goes up. This might be the growth of a plant. If you reversed it and you put independent variable first, blank depends on blank. If you said time depends on height, that doesn't make any sense. Time does not depend on height. And so you know that you've reversed them and you didn't correctly identify that variable. It always will be blank depends on blank. Okay, so back to parts of an experiment. So I said that the experimental group is the group that is exposed to the independent variable. The control group is the group that is not exposed to the independent variable. The control group is treated just like the experimental group, except they don't have the effect of the independent variable. And the only reason for having a group that is not exposed to the independent variable is to be able to compare the two groups. The last part of an experiment that is essential is to have constants. You've got to have some things in the control group and the experimental group that are the same. I'll give you a couple examples in just a second. All right, so here's an example where we have 2 plots of land. On each plot, there are 3 rows of plants. The control group, remember, is the group that is not exposed to the independent variable, has soil, water, and sunlight. I always like to find the constants first. So basically look at the 2 and what do they have in common? Well, they have 3 rows of plants. They each do. And then they each also have soil, water, and sunlight. For purposes of this, we're going to assume that it's the same soil. They're receiving the same amount of water and the same amount of sunlight. Now, to figure out what the independent variable is, you look at what's different about the 2. And the variable group, which is just another name for experimental group, has fertilizer, and the control group does not. So the independent variable is fertilizer. The dependent variable is a little harder to figure out because it's not on here. You can't actually see it, but you can ask yourself, why would they be giving one set of plants fertilizer and not another? Well, the most likely reason would be to figure out if the plants will grow better. So plant growth is the dependent variable. What is being measured? The growth of the plants. Here's another example of experimental design. So you have 2 groups, experimental group and the control group. You have a bunch of seasick sailors. The experimental group is given fruit to see if fruit helps them with their seasickness, while the control group is not given anything. The constants in this one are a little bit hard to see because you can't actually see them here, but we know that they're on the same boat. So that's a constant. They're on the same boat. The dependent variable is what's being measured. So why are the seasick sailors given fruit? To see if it affects the level of seasickness. So the thing that's being measured in this is how sick are the sailors. This is just another example of how to set up an experiment. And this is pretty similar to the other one, but I'm going to go through it anyways because sometimes understanding how to choose an independent variable and a dependent variable can be kind of tricky. So this one's a little similar in that we're using plants. You wonder what the effect soil type has on plant growth, since your plant in clay is not growing too well. You get another plant that's exactly the same type and size and use sand instead of clay. All right, so I just used a special word there, same. The word same tells me that those are constants. Same what? Type and size. So now I'm changing the soil type. The thing that I changed, that's the independent variable. So soil type is the independent variable. Since this whole purpose of the experiment is to figure out how soil type affects the growth is the dependent variable. It's what I'm measuring. So which plant is the control? The plant with the sand or the clay? What are some constants? If you are still having some trouble understanding the difference, it's okay. We're going to be doing lots of practice, and don't forget to use the lesson request board. Have a great day.